Welcome everyone to week number 4. Can you believe it? We're almost halfway through this term. So you're almost there. Let's keep going. Keep on track. Here's one of my pictures again. You know me, I'm always putting my pictures in here of a sunset at Seabrook Island. Oh, I wish I were there. And I could teach from there. I really could. But so far I haven't had a chance to go. And there's a seagull. Okay, so let's get started with this week. This week is all about cellular respiration and photosynthesis, and we're going to talk about that in a second. Let's recap last week first of all. So diffusion. Diffusion is movement of molecules from areas of high concentration to lower concentration until equilibrium is reached. And a particular type of diffusion is where water moves across a semi-permeable membrane. That is called osmosis. And so you looked at osmosis with the dialysis tubing experiment. You looked at diffusion with the starch and the iodine solution, starch in the potatoes and the iodine solution. But you were also trying to calculate surface area compared to volume in cells with that particular experiment. You did, you produced some cells in a virus just by your drawings, and then you tried to identify them, or you did identify them with your dichotomous keys. So you learned about cells, bacterial cells, and eukaryotic cells like our cells. And you also learned about viruses, which of course are non-living because I told you that last week. I did talk last week about the coronavirus that's going around, making its way around the world right now. And I haven't gotten any questions yet, but if you do have any questions, please let me know. It's kind of interesting that they brought up a couple of drugs that might be used in the treatment of this particular disease, COVID-19. The one that's used for malaria, I see no way in this world would that be effective because it affects the hemoglobin in your red blood cells, and this virus attacks respiratory tract cells. So I have no idea how that would work. I don't see how it could. So if you have any questions like that, please feel free to contact me. And I'll be glad to look up any information you need looked up or explain things to you. Just ask. So that was last week. This week, we're learning about cellular respiration and photosynthesis. So what is cellular respiration? Well, think about it for a second. What is respiration? You already know what respiration is. You breathe in and you breathe out. That's respiration. So what are you doing when you breathe in and out? You breathe in to pull oxygen into your body for your cells to use, and you breathe out to release carbon dioxide as a waste product. So the reason that you breathe in and out is so that you deliver oxygen to your cells, cellular respiration. And that oxygen is used to produce ATP, which is adenosine triphosphate, the most important energy molecule in cells, and that is made from food. But you have to have oxygen present, especially in large organisms, in order for ATP to be produced. And what's the waste product of cellular respiration but carbon dioxide? We'll also touch on photosynthesis, though you don't have an experiment about it. And photosynthesis is glucose being produced from the use of sunlight and carbon dioxide to make the glucose, and oxygen is the waste product. So you see, it's sort of an opposite reaction of cellular respiration, but really it isn't, because plants and other photosynthesizers actually do both of these processes. They do photosynthesis to make their food, and then they break down their food through cellular respiration to make ATP. So just so you understand that, photosynthesizers do both of these, and then if you're organisms like us who have to consume other organisms as food instead of making our own, we only do cellular respiration. Okay, let's talk about the lab for this week, Lab 4. I explained last week that Lab 4 is going to take a little bit longer this week because you have 2 days of prep that you have to do before you can actually do the experiment. So day 1, and this has to be started by Thursday. No question about this. So start it now is my suggestion. Day 1, you soak some of the peas that you have in your kit, in your lab kit, overnight in unchlorinated water. So you use distilled water, water in a bottle, or some of that water that you left out for your last lab. Just leave some overnight in a big pan, and that will be perfect. You'll get rid of the chlorine in your water. So soak part of your peas, and the lab tells you exactly how many. Soak the peas overnight. So the next day you put those peas on a wet paper towel soaked with what? Unchlorinated water, and you leave those covered up overnight out of sunlight because you really don't want them producing leaves yet. You don't want them to do photosynthesis yet. So that's day 1 and day 2, and both of those processes take even overnight. So then on the 3rd day, you can actually do the experiment. You're going to set up some respirometers, and the respirometers are going to detect how much carbon dioxide is being produced, and that gives you an idea of the rate of cellular respiration. If you measure a waste product, that does give you an idea of how much cellular respiration is taking place. And you will see how to do that. You'll put some food coloring, and feel free to use the red food coloring from last week at in the end of the respirometers this week, because that works perfectly fine. So you'll watch how that yellow or red food coloring moves up your pipette tip through the process of cellular respiration. So you'll set up your respirometers and then conduct the experiment. It takes a little time to set them up, but it takes about 20 minutes to run the experiment. I want to caution you this week. You're using potassium hydroxide in the bottom of your vial that you eventually put your peas in. This is not something that little children should be around, and you should be wearing all of your safety gear for this lab because potassium hydroxide is caustic. I know this. We used to do this experiment using, instead of germinating peas, we used mice and we used lizards, anoles. Of course, we had a bigger contraption. We had big flasks for the mice and the lizards, but inevitably, some of the mice and some of the lizards would get their feet burned by the potassium hydroxide. So please, please be careful with this stuff and wear your safety equipment like you're supposed to, and then dispose of all of this correctly. Get rid of this stuff, put it in the trash can as soon as you possibly can. Don't just put it in the trash and let it sit there for some little child to come along and put their hand on. So get rid of it. You read your safety manual, and you've agreed to use safety precautions, so remember that, and please do that this week. All right. The lab also doesn't explain to you how to use the parafilm exactly. And if you don't use it correctly, then it will just slide up and down the pipette. So I made you a short video last night showing you how to use parafilm. It's a tiny, short little video, about a minute long, in the announcements, but it shows you how to use the parafilm, and this helps to form the seal between the pipette and the rubber stopper that's part of your respirometer. And if you don't do this, water will leak into the vessel, and then you have to start the whole experiment over, including what? Going all the way back to day 1, soaking those peas overnight. So you see how important it is. So do take a look at that short little video in the announcements. Other than that, that's what I have for you for this week. If you have any questions, if you're having any issues at home, because I know so many people are at home now with their kids, spouses out of work, I know how it is because we've got this in our family too. So I really do know how this is. Please just email me. Let me know what's going on. We can give you extensions on your time if you need them, but realize that at the end of the semester you need to be finished, or we need to give you an incomplete. So just realize that. But if you need time extensions, please just let me know. I can work with you on this. I understand how crazy life is right now and how hard it's going to be for you to get your work done. So just let me know. That's all you need to do. And I wanted to remind you, as I always do, that if you need help, if you have any questions about your lab reports, if you have any questions about calculations, if you have any issues that are coming up with your family where you can't get your work done, just email me, m.sigmon@snhu.edu. Please email me. I'm here for you. I can help you. If we need to have a face-to-face conversation, we can do that. We'll set something up. I've got Zoom, I've got Google Hangouts, whatever. We'll figure out something. So please just let me know if you need some help. I am here to help you. So let's have a great week. And remember, get started early.